in order to get the pure excitatory response of the cell, all the inhibitory actions in the ommatidia must be eliminated. Self-inhibition, which is the inhibitory action of the unit upon itself, can be suppressed through application of tetrodotoxin, a positive neurotoxin. However, lateral inhibition, which is the inhibitory action from other ommatidial units upon the ommatidia under investigation, is harder to suppress pharmacologically because the neurotoxin that mediates it hasn't been discovered yet. But through using a thin diameter light pipe that is positioned on top of on the unit and in line with its axis, it's possible to avoid lateral inhibition by just stimulating this unit and avoiding the stimulation of the other units. So, with the long-term goal of developing Passaglia's model to account for low light level conditions, this project is involved with the development of an experimental apparatus to simulate individual ommatidia and characterize the ommatidia's response for low ambient light level conditions. So, the system I built is presented here. It consists of 4 subsystems: a control and modeling interface, an optical apparatus, a mechanical stage, and a neurophysiologic recording equipment. The desired stimuli waveform is generated through a computer and outputted to an LED through a data acquisition board, where it is collected into a thin diameter light pipe that is positioned on the crab's eye through a special mechanical stage with 6 degrees of freedom. The neural response is recorded through a neurophysiologic recording equipment into the computer who generated the signal, forming a closed-loop system. I'll go into depth into each of the subsystems now. So, the light source for the system is a green ultra-bright LED, which was chosen because green light is the crab is most sensitive to green light. The interface was developed through recording the LED response with a digital optometer, and the resulting interface holds 2 main functions. The first one is correcting for the LED nonlinear characteristics in response to a ramp input voltage through a gamma correction method, and the second one is a model of the LED system that allows me to predict and determine the stimuli I want to present to the crab. The mechanical stage, holding 6 degrees of freedom, is composed of 2 components. The first one is a linear that positions the light pipe on top of the eye, and the second one is a radial, which was self-developed, which basically allows me to position the light pipe on top of the eye and in line with its axis. So, as you can see in the movie, the light pipe is rotated through the radial component about a point in space, remotely, which is what I used in the experiment. Okay. The optical system couples the light from the LED into a thin diameter light pipe, where it is transmitted into a single ommatidial unit in the crab's eye. The low light level reduction effects which are needed for the experiment were achieved through neutral density filter papers, which were embedded in the slit of the coupler. The neurophysiologic recording equipment consists of a microsuction electrode, which draws the optic nerve into it through a low pressure created by a syringe pump. In order to summarize the apparatus development process, I present here a picture from an experiment where the light pipe is stimulating a single cell in the crab's eye, evidenced by the green beam, and the neural response is collected through the microsuction electrodes from the thin diameter optic nerve. The stimuli that were presented in the experiment consisted of sinusoidal stimulation, Gaussian noise, and a step input plus a disturbance in the form of turning on all the light in the experimental room. All the stimuli was repeatedly presented with the level of illumination decreased between repetitions. Moving to the results, the sinusoidal stimulation obtained the ommatidial tuning curve for 5 light levels. In order to quantify the results, the tuning curves were fitted with the filter model consisting of a low-pass filter and a band-pass filter, which allowed me to have the cutoff frequencies and gains for the filters. In summary, as the light level is decreased, the band-pass filter component, which is in charge for the crab's improved time resolution, diminishes. The Gaussian noise stimuli was cross-correlated with the spiking activity that resulted from the unit, resulting in the impulse response of the unit. By taking the fast Fourier transform of the impulse response, I was able to obtain the tuning curve through a second method. And by comparing the 2 tuning curves obtained by the 2 methods for 3 cells, I can see that although for each method results tuning curves that follow the same trend, the two tuning curves do not overlap, meaning that the system holds some kind of nonlinearity within it, which might be corrected through a second-order kernel, some complex filter. The step plus disturbance stimuli allowed me to quantify the self and lateral inhibitory actions in the eye. So, upon the onset of the step, the cell fires at its maximal firing rate, which is instantaneously decreased through the process of self-inhibition. When the disturbance was introduced into the system by turning on all the lights in the room, all the omatidial units in the eye were stimulated, inhibiting the units under investigation response even more, which is basically lateral inhibition, which decreased the response even more. The results were quantified, and as you can see, as the light level is decreased, the cell's firing rate decreases as well. In summary, in this project, I developed a novel experimental apparatus to simulate individual omatidial units in any compound out. I measured the temporal frequency response of the omatidial unit in the crab's eye, and I quantified the effect of self and lateral inhibition in the omatidia. Last, I would like to thank my advisor, Christopher Kosaglia, for giving me the opportunity to work on such a great project, and all the members of the Visual Information Processing Lab for their help and support throughout the year. Questions, please?
Question. Under the conditions you're looking at, did you notice any difference in the temporal structure of the spectrum?
Yeah, definitely. Cells respond differently. It takes a long time to get a good cell.
With the human eye, the photoreceptors undergo what's called dark adaptation. And I was wondering if you also had to take that into account in your model.
Well, the compound out is much more simpler than the human eye. This is why we study it. It's not as complex, so we don't have to take those things. We don't have so many interactions within it. Yep.
Concerning the curve, we plotted the transfer function for several omatidia. That one?
Yeah.
So, did you kind of look at and quantify how omatidia, their mean response to stimuli different?
Well, what I did was that I fitted them with the model parameters, but due to the shortage of time, I'm not showing those results. I quantified it, and this is how I was able to see which filter is giving more of a component to each one of the transfer functions that I get, the tuning curves.
Do you have a plot?
Okay.
Is that a question?
Yeah. Oh,
yes.
Well, do you have the plot of that, or?
A plot, not here. Parameters, they have it in a table.
Any further questions?